Alright, so hello everyone, my name is Kyra, and I will be here to serve as a partial GSA for you guys under the guidance of Dr. Geisbert. And so some of you may or may not know me, but I am just like you, so if you have any questions, feel free to reach out to me, and I will be there to assist you. Alright, so let's get started. So we are going to review the last lecture you guys did very quickly. So you guys talked about the long history of Dev Bio, and you talked about two ideas from a historical perspective: epigenesis and preformation. So what does epigenesis mean? It's a theory that states every organism is made anew from the egg, and the contrasting idea was preformation. So what's preformation? Instead of making the organism anew, it's sort of there in a tiny form all the way from the egg. And then we had the four principles of Carl Ernst von Baer. So what are those four principles? So the timing of the features, if it's a more general feature, it will happen more earlier in development, and if it's a more specialized feature, it will happen later in development. These more specialized things build off the earlier general structures. They don't just appear out of the blue. Another one, the embryos of animals don't resemble the adult forms of other animals. And the last one, so the embryos early on look very similar to other embryos, but the embryos never look similar to the adult forms. And that might be contrary to what some of you have heard, right? Like you look like a baby fish, or you have a tail because of that. That's not why, but early on structures look very similar. And all these four ideas are really interrelated to each other. Now, because the general features appear early, then everybody looks the same early on, right? And because those more specialized structures appear later, then that's when things start to differentiate. Okay, great. So you guys also talked about fate maps and cell lineage. How would you define a fate map? Well, a fate map is a picture of the embryo and sort of what structures you're going to turn into in later times of development. One of the techniques for developing fate maps is with dyes. And then the other sort of way to look at development is with lineages. So what's a cell lineage? Basically, to sum it all up, it is the family tree of cell division. So you guys also talked a little bit about transplantations. That's how some of these fate maps were done, transplanting a section from a quail into a chick embryo. We didn't talk much about ablations, or at all, but that is sort of the same idea, just destroying a part of the embryo, and we'll see that again today. And we talked briefly about C. elegans as a model system, which I'm sure by now is everyone's favorite organism. Alright, so just to compare and contrast fate maps, this is a fate map to your left showing you different stages in development and what each part of embryo is going to become, either in the adult form or in later parts of development. And to your right is the cell lineage. And together, they are going to tell us a lot of information about what is going on in development. In some organisms, whether you become endoderm or a neuron is going to depend on where you are in the cell division, because the information is going to come from within. We'll talk about that today. Now, I did already receive a question about fate maps via email. One of you guys, a student, has stated how she understands how a fate map is formed, but questioned how they find where the cell actually is, and that's a wonderful question. So yeah, it's a beautiful story actually. This guy named John Solstein for C. elegans collected the single cell egg, and he sat down at a microscope and he watched the divisions, and then he took notes. So he did it totally visually by eye, saying this cell divides into these two cells, and he gave them all names to help keep track. But it was really an amazing undertaking, so great question. Now you could also use GFP or a fluorescent dye to inject one cell and see where the two fluorescent cells are, so the tools are a little interrelated. Invertebrates and lower animals, we'll see that this lineage is going to dominate what tissues form where, and in higher organisms like us, we're going to see that these fate maps are more important. It doesn't really matter where the cell comes from, it's just where its localization is in the embryo. So a lot of information is going to come externally, and we're going to have a name for that. That's going to be called conditional specification, but we'll get to that in a second. what we're going to start with, at the end of development, you're going to have all these specialized cells. Now, how are you going to define the cell types? So we're going to talk about unique products that define each of the cell types and specialized functions. Now, hopefully this is a review for most of you, but if not, no big deal. I'm not going to ask you to memorize this table, but it'll give you a flavor for what we're talking about. So we can think about keratinocytes, and these are the outer surface of your skin. Now they have this protein called keratin, or keratin, and it's going to give you your specialized function to protect you from drying out, protect against scratches and abrasions. You have red blood cells, your erythrocytes. They have the specialized cell product, hemoglobin, really good for carrying around oxygen, and that's what its specialized function is: transport oxygen through the body. You also have lens cells in your eye, and they have this protein called crystallin, which forms kind of a crystal-like structure and lets light pass, so the light can enter your eye, go to the back of your eye in your retina, and that's how vision occurs. You have B lymphocytes that make antibodies. So I would say their product is antibodies and their function is the immune response. T lymphocytes make cytokines. They're also cell-based immunity. Melanocytes will make melanin, which gives you pigmentation in your skin. We have stem cells like the pancreatic islet beta cells. These are the only cells in your body that make insulin, so if you have type 1 diabetes, it's most likely because these cells were destroyed mostly by your body. Your body basically saw the cells as foreign, destroyed them, and now you can't make insulin. So this is a really important disease issue. How are those cells made? How can we repurpose other cells to make this insulin and hopefully take over this function? Okay, so on and so forth. We have Leydig cells that make testosterone, chondrocytes make collagen, osteoblasts make bone. And so how many different cell types do you have in your body then? Let's see, so we have 1, 2, 3, no, I'm just kidding. So we tend to recognize about 200 or 220, depending on the number. That's the sort of well-defined distinct cell types in your body. Alright, so what are we going to talk about today is how do you make those specialized cells, and there are 2 ways to do it. I already told you this. It could come from within, information from within. We're going to call that autonomous specification, or it could come from information outside the cell, and we're going to call that conditional specification. Okay, so how do you know when something is coming from within or from without? Now, this is an embryo of a patella. It's a sea snail, and there is a specialized cell that forms outside the embryo that is the cilia. And the cilia help propel it, so it's a motile embryonic stage, and these are called trochoblast cells, and they sort of define this whole group. Okay, so do the trochoblasts know that they are supposed to get cilia and be involved in movement from something from within or something from without? Now there's 2 key experiments we're going to do that are going to tell us the difference. The first experiment is we're going to isolate it, and you can take that cell that is going to be a trochoblast, put it in a dish all by itself. Does it know it's supposed to be a trochoblast? Does it divide? Does it get the cilia? In this case, the answer is yes. It knows, in the absence of the rest of the embryo, exactly what it is supposed to do. And this means that the information is coming from something within the cell, autonomous specification. Now, the other clue that is going to tell us this is if we destroy that cell, and that is going to be the trophoblast, then no other trophoblast forms. So this is an ablation. If you ablate that cell, now there are no cilia cells. That means the information to make a ciliated cell must have been in that cell we destroyed. So it's an internal signal. So this is an example of that, a little bit different example. This is a frog embryo. There's the fertilized egg, the zygote, that's the 2-cell stage. You take a hot needle and poke one of the cells, it'll die. But that other cell goes on and makes half of a blastula and half of a neurula. So it has the information in it to make a half a frog. So that information is coming from within the cell, and the left half of the frog information was there. So when we destroyed that cell, that information was lost, and now you wind up with only half a frog. So 2 ways you show autonomous. 1 is isolation, and 2 is ablation. All right, and so here is example 3, where you can do sort of both of those things. This is that sea squirt, the tunicate. Here's a fate map. The tissue on top is going to be ectoderm, those 4 cells on top. There's 8 cells here, so if you isolate those in a dish, you get a ball of ectoderm cells, so they knew they were supposed to make ectoderm. The endoderm comes from the bottom. If you isolate those, you can get endoderm. The notochord can form and the muscle can form, so all of those tissues are autonomously specified in those cells. You'll see the nervous system doesn't form, and we'll come back to that in a second. All right, so how can I prove we're going to talk a little bit about experimentally, right? So this is one piece of evidence. Can you do something else that's even more convincing? And they did this really cool experiment where you take a needle and you kind of smush the cells of the bottom in, and now the cells divide in a different place. So normally the muscle forming region, the yellow forming region, forms only from the bottom cells, never from the top. But if you take a needle and push it in before the cell division occurs, now you can get some sort of that red material in the top 2 cells, and now you can get muscle out of that. So it really more convincingly demonstrates something in those cells. We're saying, here, make a muscle here. All right, so we now come back to the nervous system. It forms from this part of the cell, but when you isolate that cell, you don't see any neurons. You don't have any nervous system. And this tells you that this tissue is not autonomously specified. It is not something from within. So the other alternative is conditional specification. Okay, and so if you isolate it and it doesn't form, that's your clue that it's conditional specification. The second test we did is we destroyed the cell and asked, does the nervous system form even in the absence of that cell? We're not going to show you that experiment because I don't think it works, but I'll show you a similar experiment here. Here with a frog embryo, here's a frog embryo, you can do a fate map and say, this patch of red cells here is going to make something in the back. Now if we take those and destroy them, we can destroy a whole chunk of this embryo and you'd get a normal frog. So there was no information in those cells that were removed that were really required to make a frog. There was no autonomous specification, or at least not as much required. You can also take these back cells and put them in the belly, and now those cells form belly cells. So they didn't really know they were supposed to be back or belly. They were relying on clues within, where they were in the embryo, to tell them what to do. All right, here is sort of an extreme example of that. This is a sea urchin. At the 4-cell stage, if you don't do anything, you get this pretty Pluteus larva that forms. It doesn't look like an urchin yet, it's motile, then it forms an urchin later. So if you take those 4 cells and separate them, put 1 in each dish, you'll still get 4 Pluteus larvae, or 4 Plutii. So everything to make an entire urchin was inside those cells. It was conditional, but they were conditionally specified. There wasn't like, 1 quarter was here, 1 quarter there. When you separated them, they could still make the entire embryo. All right, so these are sort of simple examples, but we're going to come back to these concepts throughout. All right, so now you can ask, if you take this red tissue, it's going to form a muscle. It's sort of specified to be a muscle. You take the yellow cells here and they're going to become neurons. You can ask, what if we take a muscle cell and surround it with neuronal tissue? Can it change its mind? Is this locked in so it's always going to become a muscle? Or can we trick it, give it some signal to make it change its fate? So that experiment is diagrammed here. That's what I showed you before. We're going to take this red tissue that normally forms a muscle, surround it with neurons, and say, does it switch fate and become a neuron, or does it remain a muscle? And the answer depends on what cell you take. You can get both results. So this tells you that sometimes specification is irreversible. It knows for sure and you can't change its mind. And sometimes you can change specification. So there's another term we're going to add. So specification is what we're going to say when it knows it is supposed to be a muscle. And when it is specified irreversibly, we're going to call that determination. Okay, so we're not going to use these extensively in the class, but you'll see these in different places. So specification means it knows what it's supposed to be, and determination means it's irreversibly specified. Okay, so this is table 2.2. This is sort of a nice summary table. I'm not going to read it for you, but it talks about autonomous versus conditional. Autonomous is really, really important in invertebrates, and conditional happens more in vertebrates. Now there's a third class of specification here. It's called syncytial specification. I don't agree with this classification scheme, so we're going to ignore that for now. We'll see some syncytial when we go to Drosophila development. Alright, so now we're going to ask what makes these specialized cells different from each other. Now the one that is going to become a neuron versus the one that is going to become the muscle. And the answer is each cell contains genes that are unique. Muscle cells have muscle genes, neurons have neuronal genes, etc., etc. So muscle cells don't have neuronal genes, right? Well, actually, they do. So every cell in your body has every gene in your genome. So muscle cells have neuronal genes, they have intestinal genes. And so it's not whether the gene is there or not, it's going to be called it's going to be whether the gene is expressed, and this idea that every cell has your entire genome is called genomic equivalence. So every cell in your body has your entire genome. And how did we prove this? Well, are there exceptions? There are a couple of exceptions. They're really minor. One is red blood cells. They don't often have nuclei, so they don't have the genome in them. And in immune cells, you can get some genomic rearrangements in your antibody rearranges. Alright, so the evidence for this is animal cloning, and it's not Austin Powers' view of cloning. So what is animal cloning? And here we are talking about Dolly the sheep, and Dolly the sheep was one of the first vertebrate clones that were made. Now they took a sheep, they took an udder cell, grew it in a dish, they took an egg from a different sheep. This case it's a black-faced sheep. You can tell these two sheep apart pretty easily just by looking. They took the black-faced sheep egg and took out the DNA with a little pipette, believe it or not. They took the nuclei from the skin cell in this sheep and they transferred the nuclei from. So now all the DNA is coming from the sheep. All the other parts of the egg are coming from this sheep, and it's a skin cell. So does the skin cell have enough information to make an entirely new sheep? And the surprisingly really cool answer is yes, it does. You have to do tricks here, and one is electric current to try and kind of reset the DNA marks, but if you do it just right and you're insanely lucky and you try thousands and thousands of times, you can get one that'll work, where you get a little embryo that grows in a dish, and you can transplant that embryo into another black-faced sheep. And you can get a baby that looks like the Finn Dorset sheep. So all the DNA is coming from the skin cell, not from a sperm, not from an egg, and that DNA is an exact replica of that, of the Finn Dorset strain, which is amazing. So this is pretty crazy, right? Can we do this with humans? My guess is yes. For a long time, people were trying it with other animals. Cats, you can, if you have enough money, have your cat cloned. There's a company that'll do it. I don't know if they're still in business. They used to. You'd pay them enough money. They'll take a piece of hair or something and make a clone from your cat. Worked with dogs up until last year or two. It never worked with primates. So there was something sort of unique about primate development that was preventing it, but like I said a year or two ago, there was a big breakthrough where they cloned monkeys. So it totally works now. It's tricky. Does it work with humans? I bet it would, but we don't know anybody who has tried it yet. And this was more than 20 years ago, right? 97, so probably before most of you were born, animal cloning was developed. Now, why don't we do it with humans? There's ethical reasons. One of the main things is it's really super inefficient. It took thousands and thousands of these to get one to develop normally enough to actually be born and not be totally screwed up. So when you're dealing with sheep, most of us are willing to say that's okay, although not all of us for sure. But when you're doing them with humans, there's a little bit different ethical concerns. Also, the ability to grow embryos in a dish outside the body up until now has been really limited with humans. So in the past year, there are people pushing the envelope in this to get human embryos to develop further and further in a dish. And now there are really ethical considerations. So how long can you let a human embryo develop in a dish before it's ethically acceptable to do anymore? So definitely an interesting time scientifically and also ethically. Alright, now the second thing that you've probably heard is that Dolly was messed up, and that was sort of the prevailing wisdom for a long time. Dolly died early, died young, had arthritis, some other stuff, and we'll talk a little bit about how your DNA has certain markings on it that helps turn genes on and off. And the idea was that Dolly's markings weren't quite right, so her development was wrong. And that turns out to probably not be true. It probably was just an accident. You know, every once in a while a sheep gets arthritis, just like every once in a while a human gets arthritis. And there's lots of people who have done this since and had perfectly normal animals with normal lifespan. Alright. We'll get into this, but this is really, really cool and important because now we have the ability to take any cell from your body and we can make it into a stem cell and make liver tissue, make new neurons. So the stem cell medicine has really cutting edge changing the sort of ways we might treat disease. With people with neurodegenerative diseases, we can take their skin cells, turn them into neurons, inject them into their blood, and will they go to the right place and help to maintain their neuronal function? Sounds totally like science fiction, but it does have a basis, and we're going to talk a little bit about that in this class. It's pretty cool stuff. Alright, so if every cell in your body has your entire genome, then how do different cells become specialized? And it's all about gene expression. It's not about which genes are there, it's about which genes are on and off. Alright, so, and what are genes? You guys have all taken genetics. It's a genetic unit of inheritance. That's probably how you learned it in genetics. A piece of DNA that codes for a certain trait, and you get all sorts of like tall, short, right? They just found this gene for shyness. They would have found it earlier, but it was hiding behind some other genes. No, okay. That was a little dumb. Alright, so a more modern definition. It doesn't necessarily have a defined, easily observable trait, but it makes a protein. That's a more updated version, but still a little incomplete. It doesn't have to encode for a protein. It could encode for a piece of functional RNA, but it has some sort of functional unit of your DNA. And how big is the human genome? So about 3 billion base pairs of DNA and about 25,000 genes, so somewhere between 20,000 and 30,000. Alright, so what the pattern of genes that are on or off is going to define the tissue. Oh. Ooh, Star Wars. Okay, so alright, here is an example. This is a gene called macho, and we're back in tunicates, those tunicate muscle cells. If you have the macho gene on, you're going to be muscle, right? Appropriate name, macho. Alright, so if you look in the egg and look where that gene is, you can already see that the RNA and the protein are localized in a very specific region of the embryo. So this is macho mRNA. So that gene is at the bottom of the cell, but it's not on in other parts of the cell. It's pretty cool. And as you watch the organism divide, this is the 8 cell. It's only in cell B4.1, not in the other cells. So this shows that it correlates with muscle. And the cells that express macho are going to make muscle. So does it prove that macho makes muscle? No. Even though its name suggests that it might, you may be convinced, but you should be a little skeptical, right? How do we know for sure? Because lots of things correlate with each other that don't have this causal relationship. So what kind of evidence would you want for me to prove to you that macho can make muscle? There's two types of evidence, kind of similar to what we did before. One is we're talking about the ablation of cells. We can inhibit the gene, and if you knock out the macho gene and no muscle forms, then that's more likely that macho is causing the muscle. So that's pretty good. We're going to call that necessary. Now, is macho necessary for muscle formation? What's even better than that, though, is what if we could put the macho gene in this cell? So if we can turn the gene on over here, and now this cell makes muscle, that would be pretty convincing that macho genes or macho causes muscle. So that's a much more convincing experiment, and that we're going to call sufficient. So is macho sufficient to turn this cell into a muscle-producing cell? So these are two different things, but if you can show necessary and sufficient, that's good evidence that there is a causal relationship. So here is what we're going to do. This is a later stage. These are the cells that are going to make muscle. They express macho, and this embryo, we've managed to inhibit macho in most of the cells. I'm referring to this picture right here. Here's what we're going to do in this later stage. They express macho, and this embryo, we've managed to inhibit macho in most of the cells, the macho depleted, and we can see that in this embryo, a lot of the muscles don't form at all. So macho is necessary for muscle formation. And in this embryo, we've managed to make macho be expressed in other cells. And now you get a lot more muscle formed. So macho seems to be sufficient. Alright, so in general, when you think experimentally in biology, you should be thinking necessary and sufficient. Is it required, and can it do it on its own? Alright, so what is a gene? We've talked about the unit that produces a functional protein or RNA. And what are the parts of a gene? So what information does it contain? Here is an example of a gene. So in order to understand how this gene gets turned on or off in the right places, we need to know a little bit about all of these parts. Hopefully most of this is review, but if you have a more organismal or ecology background, some of this will be new to you. So, what happens when a gene is on? The DNA for the gene gets turned into RNA, and the RNA gets turned into protein. Now, this is the central dogma of molecular biology. If you ask what is molecular biology, it's how genes get turned into proteins, how DNA goes to proteins. That's molecular biology. Alright, the process of turning DNA into RNA is called transcription, and it's produced by an enzyme RNA polymerase. Going from RNA to protein is translation, and that's done by ribosomes. So ribosomes are just huge shaped proteins. So I would get these two terms mixed up. I mean, I kind of knew them, but I'd always say translation when I meant transcription, and transcription when I meant translation, and it makes you look awful, you know? It's hard to get in the habit of this unless you talk to each other or talk out loud, right? So I now keep it straight, and one thing that helps me keep it straight is transcription. I think of monks and medieval times copying down manuscripts, right? There was no printing press back then, so how did you copy a book? Well, a monk sat there and transcribed it, made a copy, but it's the same words, it's just a new copy. And when you go from DNA to RNA, it's the same 4 nucleotides with one small exception, you know, but you go from 4 nucleotides to 4 nucleotides, and so this is more like copying, like transcribing. But if you're a monk and you have a book that's in English or in Latin and you want to translate it into French, you have to translate it. It's a lot more work. You have to change the language. So when you go from RNA to protein, we go from a set of 4 nucleotides to a set of 20 amino acids. It's a completely different language. So that's translating, just like any language. Alright, so, and these things happen spatially. The DNA is in the nucleus, you make the RNA, it goes outside of the nucleus. Translation always happens in the cytoplasm, turns into protein, and then does the specialized function. Alright, so back to the parts of a gene, and I think this is the last thing I have for you today. The protein is the functional unit in most cases. It has a start and a stop. The start is the N-terminus because it has this amino group. The end is the C-terminus. It has this carboxy group. That protein is made by the ribosome from the RNA, so the RNA has the information that AUG is the start codon. That says start making a protein here. And this information here codes for the sequence of that protein. And then there's the stop codon, but you can see the mRNA has other things in it too. There is a part at the beginning and a part at the end that is not used to make protein. And that is where a lot of the regulation is going to be, to regulate translation. So proteins have an N and C terminus, mRNAs have a 5' end and a 3' end. The 3' in eukaryotes has a poly A tail, and the 5' end has a cap. And then there are these sequences, like I said, that are not used to make proteins. They're untranslated, UTRs. Untranslated regions. There's a 5' UTR and a 3' UTR. If you want to design a regulatory sequence, it's easy to put it here because it is not going to do anything else. It's harder to put regulatory sequences in the middle. Okay, so this mRNA that's used to make a protein comes from a pre-mRNA that has some extra sequences in it called introns that are cut out and not used through a process called mRNA splicing. And the enzyme that does this is the spliceosome. Alright, so the pre-mRNA has a couple of extra chunks, and when you go back to the DNA, it's going to have the clues where to start making the mRNA. It's going to have the exons and introns, and it's going to have where to end making the RNA, which is this poly A site. But there's additional regions on the DNA too, and one of these regions is where RNA polymerase binds. That's called the promoter. And then there are other regulatory regions that have different names. Enhancers, silencers, there's insulators, all sorts of regulatory sequences. And we'll touch on them just briefly here. Alright, so when we think about a beta globin gene, this makes beta globin, carries oxygen, has that RNA polymerase binding site at the promoter, has a site to start the mRNA, has the information for where to start the protein, has introns and exons, and has the poly A addition site. So when you look at sequences, there's a lot more going on than just the protein sequence. DNA is the genetic material. It tells you which proteins to make, but it has all the regulation. It says where to make them, when to make them. There's all this information that's part of your DNA. Alright, so that's what I have for you today. We're going to just talk, a lot about transcription and transcription factors. We'll talk throughout the course of this semester how some genes are regulated by translation and the 3' UTRs. And like I said, it's new for a lot of you, but that's sort of necessary information so we understand how the genes get regulated, so we understand how development works. Alright, have a great weekend. It's the weekend now, but Monday is a holiday, so I will see you guys on Wednesday. And if you have any questions, feel free to email me. Thank you.